Today I'm here with one of my favorite colleagues, Phil Hugenholtz from University of Queensland in Australia. And we're going to be talking a little bit about his work and about microbiome. Phil, can you tell me a little bit about your research and why it's important?
Okay, so I describe myself as a microbial diversicist. So somebody
What does
that's
that mean?
that means somebody that's interested in microbial diversity from a very fundamental point of view. For the last 25 years, I suppose, I've been trying to classify and describe the microbial world, mainly in bacteria and archaea. So first with through 16S with green genes, and then more recently through genome sequences with genome taxonomy database. A very kind of fundamental thing. Because, and I don't know why, mainly because I like to quantify stuff. I like to, and I want to know what the organisms are that we share the planet with. So just a basic uh, need for discovery and understanding that
So,
way.
so why is it important?
Why is that important?
Why is it important to understand the microbial diversity in the environment?
Because the microorganisms drive the planet basically, and if you are blind to the majority of those organisms, it's very hard to tell how they're doing that, right? And the reason, of course, we're blind to the majority of that is because most of what we know about microorganisms have been what we've been able to grow on agar plates, which is a very small fraction of what's actually out there. And now we've only had the tools in the last few decades that we can really start to look at the whole picture. So that's what this is all built on the back of. So from obviously from many important angles that are important as Earth as a life system that we know we have to understand what's driving it, and particularly in the face of changes that are coming up, understanding our microbial drivers is very important. Um, why anybody would want to classify microorganisms, people have asked me that, and it's seen as a bit of an arcane sport. But actually, it's really important. It's an underpinning for everything else on top of that. So if you want to understand microbial ecology or microbial evolution, you need to be able to articulate that accurately. And just give you one example of many, if you're looking at the wrong organism, then you can be wasting an awful lot of time, or if you because you've got sloppy definitions of what the organism is. And I'm pretty sure that judging from the uptake of these very basic classification taxonomy, it's very important. It gets used. People are using it. It's really helping um, people do their science. So for instance, with the GTDB, we have three major types of users. One is and the most obvious thing is if you are exploring the environment and you get some new sequences, the first thing you want to know is what is it? And then you want to know, is it new or have you seen it before? So you can use the GTD like a yardstick, and and people do this all the time, and they say, I have this amount of percentage of new species compared to the GTB, whatever the release is, right? So that's that's
Mhm.
that's important to be able
I
to.
do that.
Yeah, you do that. It's a very common
Yeah.
thing to do. People
Yeah.
quantify that. And then the other
Because it's
thing
cool
is
to find new things, and the scientists would like to discover new things. And so having some measure of how much newness is
exactly.
always, it makes you feel accomplished. You're like, I've
Well,
discovered new stuff.
but you can articulate
Yeah.
it. You can say, I have, you know, I've looked in our study, we looked in marsupials, and we can say actually not very new in terms of high level diversity, but over 80% of the species are new
Hmm.
within marsupials. That tells me something about the
Yeah.
ecosystem, right?
Or just how much unknown biological function is out there in the world that we just don't know.
Yeah.
And it's very exciting. Part of that, so you and I both share an interest in bacteriophages.
Yes.
Why, like, what's your interest in it, and why are they important?
Well, bacteriophages drive bacterial and archaeal populations, right? So they're very big, important in terms of vectors of lateral gene transfer. Um, and they're weapons basically that we can harness
Yeah.
if we do it right. So in clinical applications and environmental applications. And plus they're just very cool. I mean, they're not really life forms, but they're like little
But,
encoded
yeah.
missiles of
And,
destruction and
and
can do a lot of things.
and they're the most abundant
They are.
biological entity on Earth,
Yeah. I'm
right?
still not convinced about the numbers, but yeah, they're huge. They're huge numbers. And in
Yeah.
terms of like, I feel now we're getting much better handle on the diversity of bacteria and archaea. And I believe that we will be able to get a really, near saturated description of that. I'm not at all convinced that it's going to be the same story for phage. It's going to take a lot longer to
Yeah.
get that mapped out. Yeah.
And so with that, so we recently put a paper together
Hmm.
that was really interesting. There was a lot of discovery in marsupial microbial samples and things like that. What was the most interesting part of that story, I think, in your opinion?
Well, there are lots of always, I mean, the problem is there's always heaps and heaps of new things and you can't pursue them all. And so you end up, the paper's usually just a collection of anecdotes. And then, of course, you put the data out there and then other people can analyze it separately and find all sorts of new things. We found Crass very common in kangaroos. Crass have been a big story amongst the in humans, for instance. And so we kangaroos and marsupials are right down in the quite a long divergence time ago, very common there. We think we've increased the host range of those Crassipherales by looking at these in the marsupial, but not the animal host range, the bacterial host range. Yeah, yeah. In fact, we may be seeing uh, uh, endosymbiotic bacterium within a Bacteroidota
Mhm.
with the Crassipherales attached. So the old Russian doll business.
Yeah.
Um, yes, we could stop the recording. I could quickly look at the paper and find out the other major things
That's
that
right. Give me the
that
statistics
but
right now
but
from memory.
but definitely the Crassipherales was a really interesting finding. The other thing, of course, with marsupials is that just the sheer diversity of, like, although now we're getting a good handle on the bacterial diversity, the archaeal, a number of new archaeal, the number of new viruses that are coming out of that study is just overwhelming. So much
Mhm.
that can be done with that data,
Yeah. Yeah.
and all nicely mapped up now with the proximity ligation, so we know who the
Yeah.
hosts are.
So let's talk about proximity ligation a little bit. So we've done work, you've done work with Phase, with our Proximeta platform, and that was one of the unique things about this study that
Yeah,
we
yeah.
just published. Um, what was the value that Proximeta brought? Like, why was it useful to you guys?
Because it tells us who goes. I mean, one of the big gaps in metagenomics is being able to associate um, the viruses with their hosts and also plasmids and other extra chromosomal elements with their host. That's a big, long-standing problem. And to be honest, the proximity ligation I always thought was a bit fiddly and expensive. And you've really streamlined that whole operation. So now that really valuable data, which tells you those associations, is all there. And that's very powerful for many, many reasons. So, you know, if you, well, as you've shown, I mean, you showed nicely today in your talk about the fact that if you have that really comprehensive documentation of which viruses go with which bacteria, then you can start to play some nice games with controlling bacterial populations through the viruses or through their gene products. So, yeah. So, and also I have to say that the metagenomes were the best ones out of it. We did some standard ones as well, but the ones that came back from you guys were really good in that regard as well. So,
Yeah, that's been really a passion of ours, is really trying to like level up the metagenome assembly and
yeah,
metagenome discovery game. We really love just discovering stuff.
yeah,
And so yeah, so we have a common interest in that regard.
yeah.
So you're part of the team that runs GTDBK.
The database GTDB and
Yes.
the tool that allows you to classify your sequences
Right.
against GTDB. Yeah, TK. Yeah.
Right. So the database and the classification tool,
Hmm.
which is really important. If somebody watching this is interested in learning more, where would they go?
to the website. It's probably the easiest. If you just type in GTDB, in the old days you would get the guitar tuning database, but these days I think you'll get the genome taxonomy database.
Oh, you finally defeated the guitar tuning people.
We finally got there.
Yeah.
So it'll come right up. Actually, we just wrote we do the NIR database issue. So we just finished writing that. There'll be a new one with all the updates and that coming out in the next few months.
Yeah.
And there's one from 2022, which sort of tells you how things are going. And the whole point is it's supposed to be very intuitive and easy to use.
Well, we certainly used it a lot, and it's amazing. It's super good.
We're very happy. It's probably the most successful thing we've done here at ACE. And it's particularly to developers, it's very satisfying to see the community using what they're producing.
Yeah, no, and I think it's been a super useful tool. Is there anything else you want to plug? Yeah.
No, I mean, I think I would like to see proximity ligation get more and more, become more and more mainstream in the environmental work. And I know that you've worked with a lot of amazing people, but I think there's definitely more scope for people to adopt the method and use it, because there's so much information in there that can be helpful for their science.
Excellent. Well, thank you so much, Phil.
Welcome.